Hi, have you ever wondered about those little things that people call bacteria? They are so little that we can see them, but they aren't the only living things that we are unable to see. You may know them because some of them cause diseases. But once again, they are not the only microscopic things that cause diseases. So then, what are they? Bacteria are living things. That is a given. In order to understand life, people have created groups based on shared characteristics. That way, organisms that share those attributes are put within the same group. One of such classifications is that of prokaryotes and eukaryotes, which is based, among other things, on the presence of the nucleus. That is where the name comes from. Karyon means nucleus, pro, before, and eos, true. So prokaryotes have no nucleus, whereas eukaryotes do. There are other key differences such as the presence of organelles, little compartments that perform specific tasks. And there is also a difference in size. Eukaryotes are generally bigger. Basically, eukaryotes seem to be more complex, and in a very simplistic way, better. But one important thing to keep in mind is that those ideas are biased to our own perception of progress. Certainly, prokaryotes have populated this planet the longest, but that doesn't make them primitive. They are basal organisms because they appeared first, and therefore they are located at the base of this tree. They are also basal because the prokaryote lineages gave rise to the rest of the living organisms. However, as a whole, they have continued to experience change. They have continued to evolve. To wrap up, not primitive, just basal, and that can be applied to other things along the tree of life when compared to other organisms. People have tried to classify all life. One very popular system considers that there are three domains: bacteria, archaea, and eukaryota. Within this classification, there is another one that I have already showed you. There is more to know about these classifications, but for now, just have in mind that bacteria and archaea are prokaryotes, as well as monera. For now, let's just focus on one domain. Bacteria are unicellular organisms, unlike you and me, and him. We are pluricellular. Certainly, we are all made up of cells, but unicellular organisms can perform every process needed to stay alive by themselves. Bacteria also reproduce without the intervention of another individual, which is not necessarily the case for other unicellular organisms. Bacteria live everywhere, at sea, on land, even within you and all over you. Well, not only you. Many organisms host bacteria, but don't feel gross. They are not there to harm you, at least not all of them. For example, most animals can digest cellulose and other insoluble carbohydrates that come mainly from plants. But microorganisms can. They provide the necessary enzymes to break them down. Bacteria living in animals' guts allow them to digest such carbohydrates and get energy from them. In the case of plants and algae, bacteria give them a hand in acquiring nutrients, particularly nitrogen, but more than another time. The point for now is that they are everywhere, even on and inside living beings, and they are very important. Where do they come from? Well, nobody knows for sure. What we know with a high level of certainty is that they were some of the first living beings in our planet. Their ancestors appeared around 4 billion years ago, probably on the bottom of the sea, within hydrothermal vents. They alone populated the Earth for a very long time, changing the environment and experiencing change themselves, eventually giving rise to the lineages that would become the rest of the living beings. But for most of the planet's existence, it has been exclusively prokaryote. We know this because of geological evidence, experiments, fossils, and also genetic information. Particularly this last one has indicated so far that all life on Earth has the same root. We all may come from one hypothetical organism called LUCA, that stands for Last Universal Common Ancestor, of which only some general features can be assumed. Now, let's take a look at the inside of a bacterium, from the inside out. You already know that they don't have a nucleus, but they still have DNA. You know, that thing that everybody talks about. DNA is a molecule, or a group of molecules, depending on how and when you see it. But in any case, it's quite long. How long? I don't know. Very long. At least when you compare it with other molecules, like water. I would like to talk more about DNA another time, because it is really fascinating. But for now, just have in mind that it is a molecule that stores information relevant to every organism, and it can interact with other molecules, like proteins and nucleotides. Anyway, bacteria have their DNA just lying there. No nucleus, no special compartment, although the region containing the DNA is called nucleoli. Within bacteria, DNA has a particular arrangement, a closed loop. That is why it is called circular DNA, even though it may be all tangled up. But the important thing is that it forms a closed loop. That closed loop forms a chromosome, so bacteria have a single chromosome. Some of them can have extrachromosomical DNA, little loops that are called plasmids, and in one organism there can be many copies of the same plasmid. Plasmids have information that is not essential to the organism, but can provide unique capabilities to survive specific conditions, such as antibiotic resistance or new metabolic capabilities. The rest of the material enclosed within the cell is the cytoplasm, a very complex mixture of proteins, plasmids, RNA, carbohydrates, lipids, minerals, and ribosomes. Some bacteria can have specific structures, but that is the basic template. The cytoplasm can appear quite messy, but it's an organized mess because there is communication between molecules, also transport, breakdown, and synthesis. Moving now to the outermost layers, the insides of the cell are held together by the cell membrane. It's important to keep the outside separated from the inside in order to keep everything working properly, and that is what the membrane does. It is formed by the type of lipids called phospholipids. They have a phosphate on top. Phospholipids are arranged in two layers. This arrangement is important for the membrane's function because it allows it to interact with the aqueous solution in both the interior and the exterior. The cell membrane has proteins, among other things, that work as gatekeepers, controlling what goes in and out. Bacteria have another cover, the cell wall. It is made up of a rigid polymer, peptidoglycan. The cell wall is like a hardened case that endures the osmotic pressure coming from the inside. There is a classification based on the characteristics of the cell wall, and identifying the group that a bacteria belongs to can be of clinical importance, and it also helps with identifying species. That is the basic template, but there are other things that a bacterium can have, like a flagellum, or multiple ones. In that case, they are called flagella. Using flagella, these little cells can move, because they work as motors that perform a rotatory movement. Bacteria receive different names according to the number and arrangement of flagella. They can be monotrichous if they have only one flagellum. It is usually located in one of the extremes of the cell, so it is also called a polar cell. If they have several flagella in the same position, they are called lophotrichous, and the reasoning is similar for other names like peritrichous and amphitrichous. Bacterial cells can also have shorter appendages called fimbriae and pili. Both are shorter than flagella and lack their rotatory mechanism, but pili are generally larger and less numerous than fimbriae. Fimbriae allow bacteria to attach to different surfaces, including hosts, in case of any bacterium that needs a host, like pathogens or symbionts. On the other hand, some pili work as receptors for viruses. They can also scar for binding sites. Others help in movement by twitching mobility, and some even work interchanging genetic material. Another important aspect regarding morphology of the bacteria is the shape of the cell. You may imagine them like this. This is called a bacillus. It is rod-shaped. If it looks like a circle or a sphere, then it is a coccus. There are different variations of these too, like diplococci, streptococci, coccobacillus, diplobacilli, and so on. There are other shapes as well, vibrio, spirochete, and many more. Why so many shapes and structures? Well, bacteria live very different lifestyles. Some are pathogens, some live within animals or plants but cause no harm, some live in water, some in soil. Between and within these environments there are different lifestyles that require different combination of shapes and structures to go through their day. Now, let's tackle reproduction. As I have said, bacteria reproduce asexually. A cell duplicates its DNA, splits down the middle, and originates two daughter cells, which do the same, over and over again. The mother cell does not survive. It becomes the two other cells. So yeah, it seems quite simple. This pattern is called binary fission. But looking a little bit deeper, the mechanisms necessary to accomplish this are way more complicated. One thing to keep in mind is that the daughter cells are identical to their mother. They have the same genetic information. That is why it qualifies as asexual reproduction. There are several ways in which they can acquire new information, but it is not through the same process. So, bacteria. But how do people know all of this? Studying these fellas is complicated because they are so small. You need a microscope, but you also need a good chunk of them, because it would be very difficult to find just one. So, you need a culture of bacteria in a growth medium, where they can reproduce so many times that they form visible colonies, except they don't come along when you first culture them. You have to find a way to eliminate those organisms that you don't need and keep alive the ones you do need. Once more, you need to help them multiply so you can finally pick them up and see them. The events that have led to the knowledge that we have today regarding microbiology and the people involved is amazing and quite inspiring, to be honest. But that topic is for another day, as well as some techniques used to study them. As you can see, there is a lot to learn about these organisms, not only about the things you have just heard, but many other things: interactions, nutrient cycles, biofilms, biotechnology, evolution, and so on. I hope we can have another time to talk about it. But for now, take care.